At some point in your life, this might have been you, and this might have been a rock, and then you might have tripped over the rock. And then if you get a small cut, you heal and everything's good again. So how come you're able to heal? Well, inside of your body, you have DNA, which are instructions, and this DNA is inside of every single cell. The DNA tells your cells to do a process called mitosis, and so one cell turns to two, and then two cells can turn to four, and the cells basically regrow, and that's how you're able to heal. So how does your cells have the instructions in order to be able to heal though? So before your cells divide, they actually have to duplicate the DNA, and that process is called DNA replication. So how does DNA replication work? Well, inside of our cells is the DNA. DNA has to separate apart, and as you know, DNA is made of nucleotides, and these nucleotides are the instructions and they provide the structure for DNA. If we zoom in order to duplicate the DNA, we separate it, and then one side is the original DNA, and the red is the new DNA that you add on. So as you know, G matches to C, C to G, A to T, and T to A. And so because of that, when the DNA separates, new nucleotides are added in, and as you can see, this piece of DNA is exactly the same as this piece of DNA when you add in the nucleotides. So this process is called semiconservative replication, and so DNA is going to do that for all of the nucleotides in order to replicate. All of this has to happen before your cells divide so that every single cell has the same instructions. Now let's look at the detailed view of what DNA replication looks like. You might be wondering, what is this 5 and 3? What does that mean? Well, here's a way to think about it. When you read a book, right? The book words have, they go from left to right. And so you can imagine 5' is one end and 3' is the other end. And it's just a way for us to differentiate which direction the DNA is going to go in. So when you read a book, you read from left to right. DNA goes from 5' to 3'. So why is that important? Well, first, in order to copy the DNA, as you saw from over here, the DNA actually has to be opened up. And so how does the DNA open it up? You have Topoisomerase, which is an enzyme to unwind the DNA. So if you think about it, DNA is like coiled up, and so topoisomerase will uncoil the DNA. Now that DNA is uncoiled, you actually have to be able to separate it in order to read the instructions on the inside. So how do we separate the DNA? Topoisomerase unwinds the DNA, but we have Helicase, which is another enzyme. Helicase helps open up the DNA. And so once it's opened up, now we can get to the instructions, right? Just like this, once we open it up, we can get the instructions. All right, so once we open it up, RNA primers, which are basically RNA molecules, they're going to be able to attach onto the DNA. And why is it important that the RNA primers attach onto the DNA? It allows DNA polymerase to be able to attach onto the RNA primer to then be able to build the DNA. So remember how over here we said that A matches to T and G matches to C? So DNA polymerase would help combine these nucleotides together to help form the new DNA. But for DNA polymerase to be able to do that, it would need to attach to the RNA primers. Now, you might be wondering, how come one seems to be continuous and one over here, the top thing over here is like 2 lines? DNA polymerase can only build from 5' to 3'. Just like when we said we read words from left to right, DNA polymerase can only add in new nucleotides on the 3' end. And so here's an example of that, right? If I'm writing words, right, let's say I want to copy this instruction, right, I would have to go from left to right. Right? Nobody writes words going backwards like this. Nobody does that, right? And so DNA polymerase can only add to the 3' end. So because of that, we have something called the leading strand and a lagging strand. Leading strand means that this DNA, the new DNA being made, it could be built continuously, meaning non-stop, because DNA polymerase can keep adding bases over to the 3' end. So as the helicase keeps going over here and opening up more DNA to copy, the DNA polymerase can just keep adding new letters, new nucleotides to this side. And so this DNA would be built non-stop. But if you look at the lagging strand, right? The lagging strand, 5' is over here, 3' is over here. But we're not opening this side of the DNA. We're only opening this side of the DNA. And so because of that, the DNA polymerase, it can only add to the 3' end, but this side is not being opened up. So what will have to happen? When the helicase opens up more DNA, then you'll need another RNA primer over here to attach on, and the DNA polymerase will then go from 5' to 3' as usual. Because of that, the lagging strand is going to be built by going backwards, but it still goes from 5 to 3, and it's going to build one piece at a time. But the leading strand, it can be built continuously as you keep opening up more of the DNA, because the 3' end can just add in new bases. So these little pieces that you get are called Okazaki fragments. So every time new DNA is opened up, the DNA polymerase will attach to a new RNA primer, and it will build a new Okazaki fragment. All of these fragments are not good because that means the DNA is not connected. So at the end, we need DNA ligase to combine these Okazaki fragments into one long DNA, just like this leading strand, one continuous DNA. Okay, and the last word that we're going to add in here is 11. This is called origin of replication because this is literally it's the origin. It's where the replication is starting in the DNA. In our cells, it's actually possible to have more than one origin of replication. So maybe over here, the DNA got unwinded by topoisomerase and helicase split it apart. But over here, maybe a helicase and a topoisomerase is opening up this part to start another origin of replication. It's possible. Origin of replication just means where is the replication starting. Okay, and that's it. That's how DNA is replicated.